Isa and Depression Scanning Calorimetry.
Assalamualaikum, my name is Adila binti Mama Amir. Today I'm going to explain halal identification with my colleague Nur Aisha Talib and Nur Fahana Bohanuddin. Basically, all meats other than what have been prohibited in the Quran and Hadith, as well as from the views of ulama, are considered halal. But the status of the meat may change according to the way of preparing it. Consequently, halal meat must be obtained from the halal sources only. All land animals are halal except for pigs, dogs, and carnivorous animals that slash and kill, such as tigers, lions, bears, cats, and similar animals. Animals with claws and elephants, and animals that are permissible to be exterminated in Islam, such as rats, centipedes, scorpions, and others similar. Halal meat can be defined as the meat from halal animals that have been slaughtered in accordance with Sharia law. Based on the Quran, Muslims are obligated to eat the meat of an animal upon which Allah's name has been invoked, which is invocation during slaughtering of the animals. In the issue of halal meat, adulteration is defined as the addition of undeclared substance or material so as to increase bulk product, making the product appear more valuable than it actually is. It does not only refer to the replacement of ingredients, but also to inappropriate information concerning the origin of raw material. Adulterated meat can be divided into 9 types, which is formalin meat, carcasses, mixed meat, longgong meat, fake meat, meat from wild animal, meat from diseased animal that contains zoonotic threat, proud meat, and meat containing drug residue. So what about this type of meat? First, formalin meat. It is a meat that has been immersed in a kind of chemical called formalin. Formalin is a solution in water that is usually used to treat dead bodies or carcasses. However, this formalin, if used in foodstuff, can be harmful because the formalin contains carcinogenic substances that can cause cancer. Second, mixed meat, which usually involve 2 types of animal which are expensive and halal meat, mixed with cheap and haram meat. For example, the mixing of meat between pork, beef, and pork is often done by butchers solely for the benefit of gaining extra profit because pork is cheaper than beef. Third, carcass is the term referring to animals that have died, either caused by accident or from disease. The animal dies without slaughter is usually due to 2 reasons. First, the animal dies alone without the cause of human interference, such as disease. Secondly, the animal dies because humans who do not meet the requirement of slaughters according to the Sharia. The dead carcasses or animal before being slaughtered cannot be eaten because of its non-halal status. Fourth is longgong meat. It is a meat injected with water to increase the weight of its meat. The quality of longgong meat is often reduced and can easily become rotten and contaminated with microbes that can harm humans. Fifth, fake meat, that refers to a substitution of physical characteristics, textures, and taste of meat to be a new physical texture and taste of others' meat, such as artificial beef and mutton, which are taken from rats and foxes. Sixth, meat from wild animals. It is the exotic meat from wild animal species, mostly from terrestrial mammals to birds and reptiles. It is mostly referred to a bush or game meat, such as wild boars, elks, deers, and pythons. 7. Meat from diseased animals containing zoonotic threats, such as meat from cow that suffer mad cow disease. This meat from infected animals will increase the risk of Creutzfeldt-Jakob disease. Zoonotic threats are infection or disease that can be transmitted or indirectly between animals and humans. For instance, by consuming contaminated foodstuff or through contact with infected animals. Next, proud meat that produced by dishonest manufacturers or butchers to increase sales profit. In Malaysia, venison is replaced by imported buffalo meats because the price of venison is higher than beef and buffalo meats. Although the horse meat and venison scandal did not involve the food safety or impose risks on human health, meat were being sold as misleading products that are not parallel with their actual ingredients by replacing high-quality meats with low-quality meats. Lastly, meat containing drug residue like beta-agonists, which can cause the side effect to the people who consume the meat. The side effects include irregular heart function, muscle shivering, headache, and difficulty to sleep. Regarding this issue, how do we derive the meat in order to maintain its halalness? Of course, by halal authentication for detection of meat product. One of the halal authentication is by using FTIR spectroscopy, also known as Fourier transform infrared spectroscopy. It is a technique used to measure the vibration of the bonds in molecular functional group that has been used to generate information on molecular composition and structure of various types of materials, including fats and oil. FTIR techniques and chemometric analysis can detect them and measure red fat levels in food samples. Therefore, a simple method such as Fourier transform infrared spectroscopy, especially in combination with chemometric techniques, has been developed, proposed, and used for identification of milk adulteration. Different species will produce different lipid components. Triglycerides, as lipids major component, have different substitution patterns, length, and degree of unsaturation. As a consequence, lipids extracted from the meat have specific FTIR spectra and fatty acid composition. As an example, beef sausage sold without registration code and halal certificate are common. This product is more susceptible to be adulterated. The laboratory analysis is taken into account as one effort to prevent adulteration of beef sausage with red meat. It is because the wavelength of the red meat is different with the beef sausage.
Another example of scientific technique for halal authentication are ELISA and diffraction scanning colorimetry. There is a research done by Koppelman in 2004 where soya protein were used as a meat substitution in processed meat industry to reduce cost. Soy protein may threaten soy allergic consumer and thus sandwich ELISA format were used to detect food allergen. ELISA detects protein from allergenic foods by using antibodies that specifically recognize the food proteins of interest. The tech developers produce these allergen-specific antibodies in laboratory animals by exposing them to the protein target of interest. Pressure scanning colorimetry is a technique used to detect changes in the physical and chemical properties of the material by detecting of heat changes. The figure shows the heating thermogram of ASC of butter and lard. The large range level of the arrangements and saturations level of triacylglycerol present in butter and lard results in the formation of different multiple endothermic and exothermic research done by Nur Hidayat in 2015 regarding detection of butter adulteration with lard by using diffraction scanning colorimetry method. Since butter has the similar characteristic to lard, makes lard a desirable adulterant in butter. DSC is a thermal analysis technique to measure the temperature and heat flows associated with phase transition in materials. From the measurement, it can provide both qualitative and quantitative information that can give us the data about endothermic and exothermic processes. The other methods in halal authentication is by using electric nose, INOS, and polymerase chain reaction PCR. INOS is a combination of electronic chemical detector series and pattern identification system that is used to identify illegal orders. It can detect both simple or complex orders. As we all know, INOS is a simple method to identify volatile compound in food products. So the advantages are it is simple, non-complex, does not require specialized software and accessible by ordinary inspector. So, what is the principle of INOS? According to a study done by Bonoski and team in 2017, volatile chemical compounds are introduced into the sensor chamber through the sampling system. The sensor's response signal is subsequently transmitted to a computer where pattern recognition is carried out. What are the applications of INOS with respect to meat? Okay, firstly, INOS can detect spoilage of labels. It can also estimate shelf life and of course halal identification. These are some studies of INOS that have been done in the past by several researchers. Okay, now let's move on to the next method, which is a polymerase chain reaction or PCR. Okay, generally PCR is the chain action using polymerase DNA enzymes that focus on DNA fragmentation by using specific primers. PCR is also widely used to identify the identity of a substance and also used to differentiate between animal species. Next, PCR is capable of detecting a DNA even in the small quantities by applying target range to DNA mounting. Okay, now let's look at the advantages of PCR. PCR is known as highly sensitive and rapid, which it is able to detect illegal substances in a short period of time and also non-complex. Basically, there are two types of PCR. The first one is conventional PCR and the second one is real-time PCR. Let's go through one by one. For the conventional PCR, the amplified products is detected by endpoint analysis by running DNA on agarose gel after the reaction has finished. Meanwhile, real-time PCR allows the amplified products to be detected and measured as the reaction progresses in real time. There are three steps in PCR reaction. The first one is denaturation, the second one is annealing, and the third one is amplification or elongation. These are some of the studies that have been done on the halal identification by using PCR. Actually, there were a lot of studies that have been done in the past, but let's just focus on the best one. And finally, these are the conclusion for halal authentication.